So, hello everyone, and thank you for taking the time to be here today. As our host mentioned, my name is Nasia Apostolou, and as a senior scientist at Emulating, I have been focused on modeling complex cell-cell interactions that drive the leaky gut syndrome in humans. And today I'm going to talk about how we leverage the biopsy-derived colon intestine chip to, first of all, benchmark the barrier protective effect of known therapeutics, and secondly, investigate if and how LGG affects the intestinal epithelial homeostasis. So IBD is definitely a debilitating disease with increasing prevalence worldwide, and especially and unfortunately in the pediatric population. But almost 50% of the patients fail to respond to the existing therapeutics. So the first question that comes in mind is, where did those therapeutics come from? And we know that the cornerstone preclinical model in IBD research is animal models. And they have definitely contributed enough. There are cases, for example, like TNF-alpha, that was a linear case translating Bent's findings into clinical bedside and have very effective therapeutic not only for IBD but also for arthritis, but that's not the case, always the case. And most of the times the species differences preclude the translation of Bent's findings into the clinical trials. So over the years, to complement those models, we came up with more human-relevant models, starting with cancer-derived cell lines, which have extensively been used in drug validation and toxicity studies. However, coming from a cancer tissue, first of all, they carry mutations, and secondly, they do not represent the cellular complexity of the healthy tissue. On the other end, organoids, they do resonate the cellular composition of the native tissue. They do maintain the unique genetic background of each patient, and theoretically they can be expanded, propagated indefinitely without carrying karyotypic changes. But they do still lack the mechanical forces that the cells sense in their native environment. And this is the gap that all the microphysiological systems try to bridge. In our case, we coupled biopsy-derived adult organoids with the organ-on-chip technology to generate the colon intestine chip. So in order to generate the intestine chip, organoids are fragmented and introduced in the top channel of the chip, juxtaposed to tissue-specific microvascular endothelial cells that line up the bottom channel. We know that the colon intestine chip allows them to expand into a confluent monolayer with site architecture, but also transcriptomic profile reminiscent of the native tissue. And for those who are not familiar with the interferon gamma triggered barrier disruption in the chip, I'm going to give a little bit of information as a background. So we are all aware of the fact that the intestinal epithelial homeostasis is shaped by a very complex cascade of cytokines secreted by both the intestinal epithelium as well as the surrounding stromal and immune cells. In our case, we decided to perturbate the colon intestine chip with interferon gamma, which is a hallmark cytokine in disrupting the epithelial barrier. Interferon gamma disrupts the barrier via two distinct mechanisms. The first one is the induction of apoptosis with parallel inhibition of proliferation, and the second one is the degeneration of the tight junctions. What we saw is that the colon intestine chip can recapitulate both of those mechanisms of action. As you can see on the left-hand side, we do observe a gradual internalization of claudin-4, occludin, and ecadherin, and relocalization of ZO1 and effactin as the concentration of interferon gamma increases. And as you can see on the right-hand side, the decreasing confluency of the epithelial monolayer and increasing cleavage of caspase-3 indicate activation of apoptosis. Moreover, the chip allows for sampling of effluent media from both channels at several time points, and what you see in this slide is, first of all, that the colon intestine chip captures the secretion of all the pro-inflammatory molecules that characterize the acute inflammatory response of intestinal mucosa. Secondly, it does capture the polarized secretion of those cytokines, and this is shown in the example of IL-6 and VCAM, which are predominantly secreted basolaterally as opposed to the apical secretion of ICAM-1. But most importantly, it captures the inter-individual variability observed in clinic, and that is evident, for example, in many of the chemokine secretion. So having that in mind, Emulate has been recruiting and phenotyping several colonic organoid donors over the years, trying to incorporate patients from various demographic, ethnographic, sex, and age background. And then we couple this phenotyping with a transcriptome profile analysis, and being aware of the rules of pharmacokinetics and having a known concentration of our substrate, which is, in our case, is interferon gamma, we leverage the RNA-seq data to determine the expression of the receptor. So what we see here is that donors with similar expression of the interferon gamma receptor show a pretty similar time and dose response to interferon. But it only takes a slightly higher expression of the receptor to significantly increase the sensitivity. And that's why we always go back and titrate and evaluate the response of each donor to interferon gamma. And in our case, with this donor, we decided to move forward by picking up an interferon gamma dose that induces a low-grade inflammation, enough though to trigger a collapse of the epithelial barrier, which is also shown by staining against zonulocludin-1, indicating disruption of the tight junctions without, however, loss of the epithelial layer confluency. So we know that the chip responds to pathophysiological perturbations, and phenocopy is the response of the human tissue to them. What about, though, being able to recapitulate the response to standard of care therapeutics as observed in clinic? According to the interferon gamma signaling pathway, we decided to move forward with tofacitinib, which is a small molecule JAK inhibitor, FDA approved and already used in clinic as a treatment of IBD in combination or not with biologics. In our system, following the cell expansion and coating of the chip with appropriate cocktail of ECM proteins for each cell type, endothelial cells and organoids are seeded in the chip, allowed to acclimate to the mechanical forces, and then on day 5, we introduce interferon basolaterally according to the site of the cytokine receptor expression in combination or not with a therapeutic. And then we monitor daily the integrity, health, homeostasis, and functionality of the epithelial barrier, followed by several readouts at the RNA and protein expression level post-completion of the study. Similarly to the case of interferon gamma, the dosing regime of tofacitinib has to be titrated as well. And in this case, we concluded that a dose of 100 micromolar was the optimal one to achieve barrier protection in the presence of low-grade inflammation. The barrier protective effect of tofacitinib was evident not only on the epithelial barrier, but also on the endothelial barrier, as shown by staining against specific proteins for each cell type. And lastly, tofacitinib modulated the secretion of 3 of the interferon gamma-induced pro-inflammatory molecules. So we have benchmarked the protective effect of a therapeutic, and then the next question is, can we assess the protective effect of a broadly used probiotic? And why that? It's known that intestine is being constantly exposed to a large microbial community and has developed appropriate homeostatic mechanism to cope with this. We also now know that that the intestinal mucosa has developed tolerance towards a part of this population, which is so-called commensal bacteria. Specific commensal bacterial strains have gained also interest over the last 2 decades, and recently many candidate drugs targeting the microbiome are already in clinical trials, and many more are being used as prophylactic agents to balance a dysbiotic gut environment. One of them is Lactobacillus rhamnosus, GG, or otherwise LGG for short, which has prominent prophylactic effects in IBD. Past studies in human-based 2D models have unraveled that 2 of LGG secreted proteins are responsible for targeting the PI3K ACT axis and restoring the epithelial junction proteins expression. So, using our interferon gamma treatment model, we start pre-acclimating the chip to LGG conditioned medium starting on day 4, and 24 hours later, we inoculate LGG. We inoculate actually 10^2 colony-forming units of LGG and allow the system to co-culture and interact for 3 days. So now many of you may be wondering why using a conditioned medium. So here we go. By inoculating LGG directly to the chip, not only we didn't see protection against the barrier collapse, but we did see that LGG triggered a loss of an increase in the epithelial apparent permeability even under baseline conditions. So we took a step back and then we tested, what about the conditioned medium alone? And the conditioned medium alone did offer protection. And then we thought, what about coupling both of them together? And it apparently did work. We achieved a maintenance of the co-culture of the colon chip with LGG for 3 days, and we also achieved barrier protection. The barrier protection was also evident again at the tight junction level, protein expression level. And lastly, we did see normalization of the pro-inflammatory cytokine secretion levels to levels similar to the therapeutic, IBD therapeutic. So to sum up, the colon intestine chip supports the ex vivo culture of human biopsy-derived organoids, and it allows them to form a monolayer with cytoarchitecture and transcriptomic profile similar, reminiscent to the human tissue. It does respond to interferon gamma, a prototype cytokine on intestinal epithelial barrier disruption in a concentration, time, and donor-dependent manner, and it captures hallmark responses to existing IBD therapeutics. And lastly, it recapitulates the anti-inflammatory effect of LGG. And with that, we have reached to the end of my presentation. I would like to say a big thank you to all my current and past colleagues that contributed to this work and brought it to fruition. And I'm happy to answer any questions you may have.
Thanks, Nasia. Beautiful data. Questions?
So hi, over here. Thank you for your talk. I was wondering, what's your hypothesis when you add the LGG? How would the effect change when the entire microbiome is also present and not just the bacteria alone? So in a real patient, basically.
Yeah, we haven't added the entire microbiome.
Yeah, sure.
We have only tested a single strain. It is known that the proteins, that there are at least 2 secreted proteins that confer protection towards the barrier disruption, and this is what we see with the protection when we use the conditioned medium. Now when it comes to why the LGG has to be combined with the conditioned medium alone, we do have a theory and a hypothesis on why this happens. The system has not reached the full complexity. We lack the innate immune cells. So we miss the tolerance and control that the innate immune cells confer towards the overgrowth of commensal bacteria. And we also know that the organoid growth medium has a very high content in glucose, which is a substrate for any bacterial strain. So by inoculating the LGG in preconditioned medium, it means that the majority of the glucose content has already been consumed, so it gives us a longer time window to experiment and prevent the overgrowth and maintain the culture for longer to see the protective effect.
Okay, thank you.
Other questions, comments?
Hi, thanks for a really nice talk. I'm just wondering what you see in terms of mucus on your model and how the bacteria is interacting with the mucus.
So that's something that we haven't studied yet, but we are actively working on optimizing the secretory lineage differentiation on the chip. We have recently achieved differentiation of goblet cells to a percent that's close to what we see in the human tissue based on the region from which the organoids have been isolated. And ultimately, that's a very good point. It is in our plans to analyze quantitatively and qualitatively the mucus barrier in our system.
So maybe one last quick question. Thank you for your talk. Thanks for your lactobacilli data. I was wondering, so here I am. So can you do you already have a suspicion in terms of the mechanisms behind the lactobacilli? Is there any change in transcriptomics or metabolomics? So
we haven't done any of this analysis yet. We are planning to, but we are still quite early in our research with such type of therapeutics. And to answer your questions, yes, some of the therapeutics we validate in the system have an already known mechanism of action. Some others, they don't. And one of the examples was, for example, cytokine L22, which based on most of the data we have so far, it is known to have a protective effect, but we did find out a different mechanism of action, which seemed to be more disruptive. So there is not always a hypothesis, but yes, in order to validate and make the system robust, most of the times we do use molecules with one at least known mechanism of action to be able to benchmark.
thank you.
I saw one question here. If it's a very short question and a very short answer, then we still have time.
Quick question. Was the colon chip under anaerobic conditions or aerobic?
Aerobic LGG is, yeah, aerobic.
Thanks. That was indeed a short question, short answer. Thanks again.